In this video we will discuss about the CAR T cells derived FASL genes regulates the cellular persistence in vivo. So in the previous video we have understood that CAR T cells regulates this FASL signaling pathways, and they wanted to see how it will affect in the in vivo studies. So in the mouse they took two kind of CAR T cells, one which does not have this FAS signalings intercellular domain and one that has the FAS signaling in domains, and they transduce the 1:1 co-infuse these 1:1 cells into the mice and observe them for this 4 week. And what they are able to see is that in the case of these CD8 plus cells, the TCM cells and this TSM cells is almost negligible in all the cases. That is the cells that has the FAS signalings, and cells that does not have the FAS signalings seems to be comparable in both the CD8 plus cells and CD4 plus cells. It can be seen here. And when they check using the flow cytometry, they can see that at the day 0 the TEGFR and TLNGFR are 1:1, but after day 27 they can see that their populations goes down. That is TLNGFR population goes down significantly in the blood, bone marrow, liver and spleen. And when they did the median analysis, here also they can see that the ratio of this TLNGFR and TEGFR by TLNGFR, in the day 0 it's almost equal to 1, but at day 27 at different locations we can see that the ratio is significantly greater than 1, indicating that these TEGFR cells are able to survive, whereas TLNGFR is not able to survive because it can trigger apoptosis through FAS signaling pathways. That is what they have found out in the previous studies that we have discussed in our previous videos. Thereafter, they further wanted to check the presence of this FASL, so they used the cell lysate and did the Western blot, and they are able to see that in the case where FASL has been knocked out, they do not see any signals, and when it is not activated, they also cells are not activated through anti-CD3 or CD28, they do not see any signals. But for the control where FASLG is not knocked out and the cells are activated, T cells are activated through anti-CD3 and CD28, they are able to see two bands at around 37 and 25 kilodaltons that could correspond to FASL genes and FASL ligands. So this indicates that the activated T cells is able to generate or will have this, or we can say that the TLNGFR cells, when they are activated, they can see the FASL ligand and FASL genes when they are activated. However, in the case where FAS is knocked out, they do not see those signals. Indicates that it's the activations that cause triggering of these signals. And when they check the ratio of the this FASL genes and FASL knockdown at different time intervals, they can see that when the FASL gene is present in the wild type case, they are able to see its signal goes up to the when they are activated, or we can say that the ratio of TEGFR and TLNGFR goes up when the cells are activated, because in the case where FASLG is been present, whereas in the case where FASLG is knocked down, they do not see any significant difference, indicating that the FASL gene might cause apoptosis, and that is the reason they are able to see less cells in the case where FASLG is present in the wild type and cells are transduced with T and GFR, that is where FAS signaling is present. And in the first case, that is on the left hand side of the panel left, the cells are activated with K562 CD19. So that's why they see a difference. When the cells are not activated, they do not see a significant difference, indicating that it's the activation that is causing the FASL signals to be get activated and trigger apoptosis. Now to further confirm these things, they use the different cell lines, or they use the same mice, but they transduce the T cells. They transduce the T cells with different CAR T cells with different CAR T, that is TLNGFR and TEGFR, and plus minus FASLG knockdown. Or they did the same studies, that is they co-transfuse them at 1:1 and check the cells at after 4 weeks. What they are able to see, that in the case of these pre-infusions, they are able to see the TEGFR and TLNGFR are present in the almost at the same level, that is 42.6 and 48%, and once they are activated, after 28 days, they are able to see only the TEGFR signals. But for the case where FASLG has been knocked down, they can see the ratios are around 44.4 and 43.9%, and this goes down to less extents, that is around 36 and 62%, only twice difference. But in the case of wild type, we can see that there is a clear removals or degradations of the cells with TL and GFR positives. And these things they have quantified in the panel I, where we can see that in the case of pre-infusion, the difference is non-significant, whereas in the case of after 28 days of the infusions, they can see the difference is significant in the where FSLG is present in the wild type, and there is no significant difference where FSLG is been knocked down when it is compared with the control. So this altogether indicates that the CAR T cells trigger FASL signaling pathways, and that can cause cellular persistence in vivo. Once the T cells, once those CAR T cells gets activated. That's what they have concluded in these results that are presented in this figure. If you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.